Yes, sir.
Okay, so for Kulsum, also we have to just know two things in my class. So first of all, my classes are very discussive. Okay, so you have to discuss, you have to be an active part of the learning. It's not going to be a one-way process. I'm not going to just tell you stories or dictate you things all the time. I will do that as well. As a teacher, I have to do that, but you have to be discussive. Okay, first thing first. Second thing, uh, about biology, the most important thing is learning uh, the how and the what part of it. And batch, I have already told everyone that how we tackle the what part is that we try to understand the terms, the words, the technical terminologies in biology. Is it? Do you understand, Kulsum?
Yeah, got it.
There are many words in biology that are very very context oriented, and whenever they come anywhere, and if you know the meaning of those words, it becomes easier for you to understand the context and also understand the process. And even if you are not aware of the process, you can have a logical guess about it, which makes it easier for you to remember biology or do good in your entrances and as well as school examinations. Okay, so you have to maintain a copy, and the first few pages of your notebook will be left empty, because there you will be making a terminology section, like just a line in between, and on one side you will write terminology and the other side you will write meaning. Is it clear also?
Okay, yes.
Yes. And with time, as we will progress, you will understand what I meant by that. Okay, so just do it, make a terminology section, terms and meaning, like this. Okay,
Okay.
is the screen visible to all of you, everyone? So since we have a new student with us, and as responsible batchmates, at least everyone should say hi to Kulsum. Just unmute yourself and say hi with your name.
Hi, Kulsum.
Hello.
Your name also, no? Kulsum does not know you.
Hi, Kulsum. I'm Noor. How are you?
I'm fine. Nice to meet you.
Nice to meet you too.
Virtual meeting. Others, just say hello and your name. I thought this generation is not shy of introducing themselves. Okay, Kulsum, so we have Arpit in the batch. Arpit, would you like to say hi to Kulsum?
Hi.
Yes.
Hello.
You all need a catalyst, you know. So you are the elements which will not react unless and until there is a catalyst. Fatima has already introduced herself, right? Fatima?
Yes, I have.
Yes. Hafsa?
Hi, Kulsum.
And we have Mehraj. Mehraj, are you there? Would you like to say hi?
Yes, sir. Hello, hello, Kulsum.
Hello.
You have having a couple of more, like 2 or 3 more batchmates who have not joined today or will join in some time. Okay. So for Kulsum, we'll just go through a brief recap, and I will be giving this. So mostly I ask students to give this, I ask questions and I ask them to give this recap most of the time. So what we did in the last class, but today I'll be doing it. Okay. Is it?
Okay.
Okay. So just a quick outlook, Kulsum. So we have started the first chapter, which is basically reproduction in organisms. But apart from that, we also have discussed certain fundamentals of biology, and all our classes are recorded. So you would like to reach back to the operations team and ask for these recordings, okay? And see for yourself, so that you do not miss whatever happened in the classes. And still, if you have doubts, and still if you need to clarify something, we will have backup classes. Is it okay? Is it cool?
Yes. Thank you.
Very good. So we started with reproduction, and we have discussed about certain fundamentals that I would first appreciate if you go back and look at the videos, and then we'll discuss about it. But the thing that you have to know, the context to stay connected in today's class is that we have started types of reproduction. So there are two types of reproduction, asexual and sexual, right? You already know about it from your junior classes, correct?
Yes.
In asexual reproduction, the most important thing is that there is no gamete fusion. There may or may not be gamete formation, which is called gametogenesis, but gamete fusion is strictly absent in asexual reproduction, and only a single sex or a single parent, as we call it, is involved. Whereas in a sexual reproduction, gamete fusion is the criteria to say that this reproduction is sexual, and two sexes are required and their gametes are also required. So these gametes are formed by these different sexes. Now both the sexes can be present in the same body, or they can be present in different bodies, right? Like in case of earthworm, which are hermaphrodites, both the sexes, both the sexual organs are present in the same body, testes and ovaries. Whereas in case of humans, we have different separate bodies for different sexualities. Is it clear till now?
Yes, got it.
So we have started with the asexual part, and especially in the lower organisms, we have discussed all the modes which are there present in the lower organisms, like binary fission, budding, fragmentation, regeneration, and vegetative propagation. From that, binary fission, budding, fragmentation, and regeneration we have done, and spore formation. So you might want to write down these things that we have already done. So go back and read about it from the NCERT first. Okay. First, make a foundation, and then anyways, I'll be teaching you this again in detail so you can clarify your doubts there. Okay.
Okay.
So till spore formation, which means budding, fission, fragmentation, regeneration, and spore formation, these 5 modes we have studied for lower organisms as an asexual mode. And for higher organisms, the asexual mode, higher organisms means higher plants, that asexual mode is called vegetative propagation. Okay. So we have studied vegetative propagation. And in vegetative propagation, in the last class we did this. There are 2 ways of doing vegetative propagation. One is natural and the other is artificial. Okay. Now vegetative propagation is simply any vegetative part of the plant, a higher plant. For example, either it is stem, leaves, or roots. These are 3 vegetative parts. Okay. Flowers and fruits are regenerative or sexual parts. So any vegetative part can be used to propagate the plant without any gamete formation. So that is called a vegetative propagation. It is an asexual mode. So in this, we covered that naturally it can occur through leaves, like in case of bryophyllum, which is, again, like this is just a recap. We will be doing this in detail later on. But just know that through leaves, in bryophyllum it happens. Bryophyllum has small buds at the notch of the leaves, which when fall on the ground in the soil, develop into a new plant. Okay. It can also happen through stem, where these stems are specialized adventitious stems and they are called runners. Okay. Through roots, it happens through tubers. Now tubers are of 2 types, root tubers and also stem tubers. So potato is a stem, right? You know that?
Yes, yes.
It's a modified adventitious stem, whereas sweet potato is a modified adventitious root. So through that also plants can propagate without making seeds or any other kind of gamete formation. And the 4th thing that we studied was bulbs. They're also known as bulbils. Sometimes they are written as bulbils in NCERT. And about bulbs, the example is of onion and garlic. So you know that in onion and garlic, the edible part actually is a leaf, right?
Yes.
And this here, if you can see, they have a very reduced discoid stem. Okay. This part here. So this stem is very reduced, and from the stem, small roots come out. So this stem is mostly in the soil, and the root is also obviously in the soil, and on the stem is attached the leaves directly. So no concept of branches are here. Okay, in this plant. So there is a stem, which is a reduced disc-like stem, and then on which there is a bud in the center. So if you see an onion or a garlic, in the center you will find there is a bud when it is very, very young. If you see a young onion, and all these edible parts in garlic or the layers of the onion, they are actually modified fleshy leaves. Okay. So this structure where on a stem there are leaves attached, and those leaves are edible, containing in the center buds, is called a bulbil or a bulb. Okay. It's called a bulb because a bulb looks like this. So bulb has, if you see a conventional bulb, it has a filament in the middle, and then it is covered by a glass, right? And it glows. That's why it's called a bulb. The arrangement is same. And inside the bulb, in the center, there are two types of bud: apical bud and lateral bud. Apical bud gives rise to leaves and flowers, and lateral buds give rise to the shoot, which means stem and other branches, if that plant has branches. Is it clear till here?
Yes, got it.
Yes. And today we will be starting with the artificial mode, right, everyone? This is where we were, right, Fatima?
Yes, sir.
Yes. So today we'll be starting with the artificial mode of vegetative propagation, and this is the mode which actually is discussed in most of the books, and we think is the way to vegetatively propagate plants. But this is artificial. Okay. So natural is where there is no human interference. So in all the examples above, whether it was happening through leaves, through stem, runner stems, through tubers, in roots, or through bulbs, it was happening on its own. No human interference was required. But in cutting, grafting, layering, and tissue culture, which I will be teaching you today, in today's class we'll cover these topics. They are done by humans and for the benefit of human through these plants. Is it okay? So let's start with the artificial mode. And the first one is cutting. Anyone has any doubts before I begin the class? Anything you want to ask me? Biology, non-biology, anything to discuss? No? Okay, so let's begin. So the first thing that we'll be talking about today is cutting. And as the term suggests, it's simple. Cutting means you are just cutting any vegetative part of a plant and putting it directly in the soil. Is it clear? So just cut something and put it in the soil, and that part which we have cut is capable of giving rise to the whole plant. Now, I have told you before also, and this is for Kulsum again, that vegetative propagation, it happens in higher organisms, like in higher plants. That's why we study it as a separate mode. But basically, if you go to the fundamentals of what is happening, then the biology is simple. The biology is just regeneration or fragmentation, correct?
Yes,
So
sir.
in natural mode of vegetative propagation, isn't it just equivalent to a regeneration? So what used to happen in regeneration, Fatima? Remember regeneration? I gave you the example of lizard. What was regeneration?
The lizard grows back its tail.
Exactly. So the ability to grow back the lost body parts, right? That is regeneration. Or in case of some organisms like planaria and hydra, it is also the ability to regrow the whole organism from a lost body part, correct?
Yes, sir.
Right? Yes. So this is exactly what is happening here in plants. If it is a leaf containing a small bud-like structure at the notch, or if it is a runner. So if a plant is generating an additional stem which is growing horizontal to the ground, isn't it just growing something, like regenerating something? So this plant is regenerated back from this runner, correct? Do you understand the concept? Active cell division and making new structures. Make sense or no?
Yes.
So biologically, it's the same process happening in higher plants, not in lower, simpler organisms. So we call it is vegetative propagation. Okay, whether it's tuber, whether it's bulb, every structure is regenerating other structures and finally regenerating the whole new plant. Okay. In case of artificial modes, it's fragmentation followed by regeneration. And what was fragmentation this time? Arpit, fragmentation.
Where it's when the parent body is broken down into fragments and then each fragment is individually grown.
Exactly. And for this breaking down into fragments, what was required?
So there is a requirement of an external factor.
An external factor was required, right? So if a hydra reproduces through fragmentation, it has to depend on an external factor to get fragmented first, correct?
Yes, sir.
And then if the fragments, if we have fragmented it into 3 parts, then all the 3 parts will develop into 3 new hydra, correct? This is what the logic was.
Yes, sir.
Same logic applies here. You are fragmenting a higher plant here. You are cutting it. So you are cutting. So the first process, which is cutting, in this you are cutting apart either a stem, and when we are cutting it, we only can cut a stem or a leaf. Cutting a root and putting the root in the soil does not make sense, correct? Because the root is already in the soil. Yes or no? You understand what I'm trying to say? When we are cutting some part, then we are putting it in the soil for its growth. Yes, Kulsu?
Yes, I understand.
And the root is already in the soil, correct?
Yes.
So for cutting, what we can exploit are the stems and the leaves. Okay. So write down cutting. Let's write down the definition for it. In the process, in cutting process, a part of plant, specifically a stem or a leaf, part of plant, specifically a stem or a leaf, is cut and planted in the soil. Stem or a leaf is cut and planted in the soil. Next point, these cuttings sometimes require plant growth hormones. Remember plant growth hormones, class 11th?
Yes, sir.
Plant growth regulators, yes. So we treat them with plant growth hormones so that they can start inducing the structures which are not there. So if I cut a stem, so for stem an example is rose. Rose plant can be propagated through cutting. So if you see gardeners propagating rose, they cut a branch of the rose and then they put it in the soil, and then they give it like nutrition, give them nutrition, water, and sometimes also some plant growth hormones that allows that stem to now start growing roots. Okay. Remember class 11th, you must have studied that there are many hormones which start fruit formation, root formation, shoot formation, ripening of fruit, etc.
Yes, sir.
Yes. So write down. Require plant growth hormones to induce root development. To induce root development. Okay. Now, even if we give plant growth hormones to the cut part, and let's say it starts developing roots, but normally, did you expect roots to start growing from a branch, a cut branch, or a leaf naturally, like in normal ways? Yes or no?
No.
No, right? So in biology, when structures like roots, stems, or leaves are produced or are generated in places where they should not have been generated normally, naturally, are called adventitious roots. Adventitious roots and adventitious shoots, branch or stem or leaf, anything, okay? And it gives some advantage to the plant. The advantage which would not have been there if these structures were not developed. All of you have seen banyan tree, a banyan tree? Yes or no?
Yes, sir.
Banyan tree. And in a banyan tree, you will see structures coming down from the branch towards the ground. Have you seen that? Yes or no?
Yes, sir.
What are those structures? Can anyone tell me? So banyan tree is a big tree, which is like very it has branches.
Stems.
From branches, we think that things like this come down, right?
Yes, sir.
What are these structures which come down? Also? What are these?
Modified leaves.
No, they are actually modified roots. They are adventitious roots. Now, this is very interesting because roots are not supposed to be growing from the branches down towards the ground, correct?
Yes, sir.
Roots have to be down inside the ground. So they are called, so they are present at a place where they normally should not have been present, but their presence is giving the plant some very important advantages. So it's called adventitious structure. So this is called adventitious root. You also might have seen a sugar cane. Anyone has seen a sugar cane?
Yes, sir.
Yes.
So a sugar cane looks like it comes segmented. So the sugar cane body is segmented like this, right?
Yeah.
There are different segments. And the sugar cane which we get like from the market is very processed and cleaned. But if you see a sugar cane in the field, you will see that from these nodes, so these are called nodes. From these nodes, there are certain structures like this coming out, right? See this? It needs to be cleaned. You know, what are these structures?
Leaves.
What?
Leaves.
Leaves. No, they are also modified roots. Okay. So if you cut a sugar cane from here and put it in the soil, it will propagate into another sugar cane. Is it clear? Makes sense? Is it clear, everyone? What are adventitious structures?
Yes.
Okay. So write down. After treating with plant growth hormones, new structures like adventitious roots develop from the cutting. New structures like adventitious roots develop from the cutting. So cutting is very simple, as the name suggests. You are cutting something, putting it in the soil, right? No issues.
Sir, the banyan tree, but that is not artificial.
What?
That is not artificial. Banyan tree.
Banyan tree.
Yeah. The modified roots on the banyan tree, they are not that is not they didn't come because of artificial mode, right?
Yes, right. You mean to say that they didn't come because we treated them with plant growth hormones or something like that. They are
Yeah.
naturally came. Okay, yes, you are correct. So as I told you, adventitious structures can naturally also form, right? Because vegetative propagation naturally also happens, no? Even in sugarcane, these structures come naturally. But when we are utilizing these knowledge, so we have knowledge that there are adventitious structures possible in these plants. So we have a list of plants in which adventitious structures are possible. So what we do is we just utilize those adventitious structures by artificially propagating these structures. So we know that a rose plant is capable of forming adventitious roots right from its branch. So we cut the rose branch and put it in the soil, and our problem is sorted. We get another rose plant. Do you understand, Fatima?
Yes, sir.
Yes, makes sense to you now?
Yeah.
Okay. Second thing is grafting. And how is grafting different from cutting? Can anyone tell me what is grafting?
In grafting, you put this, like you join parts of the two different trees.
Different plants, yes. So grafting is cutting. Okay, it's first we are fragmenting. So first step is cutting only, but not putting it in soil, but putting it on another plant. So taking a tissue from one plant and putting it on the another plant is called grafting. Is it clear?
Yes, sir.
Yes. Just like we also do grafting for like humans and animals, skin grafting, we call it plastic surgery sometimes. Have you heard the term plastic surgery? Facelift surgery. Somewhere if you get burns on the skin, so you can take skin from some other part of the body and graft it on some other region where it is required. You understand? That is a surgical grafting procedure. In plants, we are doing the same thing, but in a less complicated manner. Very simply, we are just cutting a part of plant and putting it, because plants, they do not die like animals. They do die, but the death there is not like animals. Their organ system level of organization is also different. Remember class 11? Cellular organization, tissue, organ system. Yes or no?
Yes.
Yes. Perfect. So write down grafting. Grafting. Let's first define it. In grafting, the cutting from some other plant, in grafting, the cutting from some other plant is attached to the stem of another rooted plant. Another rooted plant. Rooted means it's already in the soil, okay? Another rooted plant. So basically what we are doing is,
Sir,
yes, Fatima.
sir, there are some fruits which are hybrid fruits, right? Like watermelon, which is yellow in color, or apple, which is different. Okay. So how is that made? Is it because of grafting?
Yes, I'll come to that. In watermelon, the concept of grafting cannot fix in, so they have to be genetically modified. So there are seedless watermelon. You know about that?
Yes, sir.
Yes. Anyone has had seedless watermelon?
Yeah.
You have had? Nice. Do they
And
taste
also yellow watermelon, like yesterday only.
yellow? From inside it is yellow, right? The reddish
Yeah.
part is yellow, actually.
Correct.
Exactly. So what makes a watermelon red?
The gene?
Pigment color.
Perfect. Pigments. What makes anything which is colorful in biology, either it's because of pigments. If it is internal, it's a pigment protein that is making it colorful, or it is some laws of physics. So many butterflies look different from different angles. The wings of butterfly, they change color when you look them from different angles. Even peacock feather. Have you seen a peacock feather?
Yeah, sir.
Turn it against the light, the color will keep changing with the angle you are changing.
Yeah.
Yes, sir.
Yes?
Yes, sir.
So there it's not pigment, because if it is a pigment will look the same from any direction because it's pigment. Blood has a pigment called hemoglobin. Okay? You know hemoglobin?
Sir.
Yes.
Gives blood the red color. So blood looks red, you look it from any angle. And if something is changing color, like a chameleon changes color, right?
Yes, sir.
It changes its color because there are certain scaly cells on the surface which actually reflect different wavelengths of light back when light falls with different angle of incidence. So either color in the natural world is coming because of biology, and if it is coming because of biology, it's coming because of pigments, or it's coming because of physics, laws of physics. In physics, you must have heard that angle of incidence is equal to angle of reflection for any surface, correct? You draw a normal. Yes or no?
Yes, sir.
And you also, so this is reflection, laws of reflection. There is something called laws of refraction, where a light, when a light travels through two different media, it bends either towards the normal or it bends away from the normal, correct?
Yes, sir.
So these are the laws of optics that helps us to see things. And if in a biological organism, the color is a result of physical laws, the possibility is it will change when you will change the falling of light and other physical parameters. But if that color is a result of pigment, it will stay the way it is. So in watermelon, the color comes from pigment. Okay. Have you seen carrots? Carrots come in all shades of orange, yellow, and red. Same is now with capsicums, right, Fatima?
I have seen orange carrots, no other color carrots,
You've
but
not-
like capsicum, yeah.
You've not seen a yellow carrot?
No.
Okay. So there are red, orange, yellow, three shades of carrot, and in that also there are different like, you know, in that yellow also has shades, orange also has shades. So different carrots species can be of different colors, ranging from dark red to very light yellow. Same is with capsicum, right? Capsicum comes in yellow, red, green.
Yeah, yes, sir.
Similarly, because the gene that is giving rise to the pigment, that gene, which pigment it is giving rise to, decides the color of that fruit. Okay. Same is with other fruits also. Guava. Some guavas are red from inside, some are whitish. Correct?
Yeah.
So it's not a very big thing to achieve in biology. It's very simple because we just need to take out some gene and put in some other gene for some other different pigment. So most of the time they are genetically modified or a different variety that we are growing. Grafting does not work much. I'm not sure if it can work in a watermelon because watermelon grow on creepers, right?
Yes.
So it does not have a very erect stem on which you can graft something very easily. But I'll try to find out how, is it possible. But I know that genetic modification is possible. But yes, the other question that you asked is possible through grafting. Like I'll be coming to that also. Actually, I was about to give that example. But let's first write grafting. Did you write grafting? Did I?
Yes, only the
No, sir.
definition.
Definition, right?
Yes.
Write down the tissues, the tissue of the graft. So the graft is this thing. So let's say there is a plant which is rooted in the soil. This is the root system of the plant, which is very good, flourishing and doing well. But you cut this plant from here, and then there is another plant whose root system is not very good. Okay, so for this plant, only the shoot system is very brilliant, but the root system is poor. Okay, it's not well developed. Do you understand? Or even if it is well developed, it can be well developed as well, but you want to make hybrids like Fatima asked. So you will cut this part, and this part will be now called a graft. Whatever we are taking, always graft is a shoot part. Remember, you can write it somewhere. Graft is always a shoot part. There is no point of making root. So if you're cutting root, you are uprooting it and then putting it somewhere else. It does not make sense. So graft is always a shoot part, which is grafted on a pre-existing rooted system. rooted plant system. Is it clear?
Yes, sir.
Understood what I'm trying to say? Now, this part, when it is grafted here, can anyone tell me what are the limitations of the grafting process? Can we do it for all plants?
So I think like how the organs reject the other organs, the plant can also reject the new plant.
Yes, right. But here do plants have an immune system?
No.
No, plants do not have an immune system. So, but you are correct. Something like that must be happening, but not exactly as a rejection, but as not able to accept. Do you understand? It will not reject it, but the point first is it is like an open wound in a plant system, correct? Because you are cutting from here, cutting from here. That's why we have to cover this part. So this patch needs to be covered so that first of all, there is no infection. Let me tell you that we always think that infection is a very animal centric thing in biology. It's not true. There are pathogens which specifically attack on plants. So plants also have their own pathogens, bacteria, fungus, and viruses. Do you know that? Very specific viruses and bacteria only infect the plants. Okay. So to avoid infection, no infection should occur. We cover it. You know, we make it hygienic, like we cover it with something which does not allow any bacterial or fungal growth around that tissue. Second thing is because there are two tissues trying to accept each other. In this case, in class 11th, have you studied if I make a cross section of a stem? Let's say this is the cross section of the stem. In a stem, there are many xylems and phloem bundles. You know that? Arranged in a specific manner. Yes, please. Who was speaking what?
I was saying yes.
Yes, right. So let's say this is the arrangement of the xylem and phloem bundle in one plant. You know that they are arranged in a stem in a specific pattern, correct? So if this is the pattern for xylem and phloem bundle, you would like this to match with the graft pattern as well, correct? Because when it will become one single plant, whatever is being transported from the root system should go all the way to the shoot system, correct? And if these xylem phloem bundles are not matched properly, this is what matching here is, Fatima. Do you understand?
Yes, sir.
If these does not correctly align, then do you think that the plant will be able to transport nutrients to the top shoot part, which is grafted? No, right? So xylem or phloem bundle should match for shoot and the root system. Okay. Additionally, also the diameter should match. Correct. This is very obvious. You cannot take a root system, a shoot system like this, and then try to put a shoot system which is of this diameter. Is it possible? No, right? The other way around, where you have this is the root system, and you want to put a shoot system like this. Do you think it will be compatible? No, right?
So in this case, the xylem and phloem only will not match, right?
Exactly. It will not match. And sometimes what we can also do is, if we have to do this kind of thing, where the shoot system is smaller, then we take the root system and instead of putting it on the top, we make a space like this, and then the shoot system from some other plant is grafted like this. Do you understand? Make a fit in the stem.
So I didn't understand this.
So this is the stem, okay, of a root system. This is clear?
Yeah.
And suppose you do not want to put another stem on this, a full-fledged stem. So you will not cut the stem part. It will be there. But instead, in this stem, you will make a cut like this. You understand?
Yeah.
And you will take a small branch from some other plant which you want to make a hybrid with, and suppose this is the stem with leaves and flowers, okay, and you place it like this on the stem. Now is it clear?
Yes, sir.
It's like on a wood, you make a indent, and in that indent, suppose this is the shoot part, in this indent you fix this so it stays there, and then you cover this part so that there is no infection. After some time, water vasculature bundle will form. It will find a way to send nutrients here, okay, and then this will start growing in width and in length. Is it making sense, Fatima?
Yes, sir.
For everyone, wholesome? Everyone?
Yes, sir.
Yes? Cool. But now tell me, first, after explaining everything, tell me what do you think are the limitations of grafting? First, complete what, okay, sorry. First, let's complete what I was dictating, or have I completed it?
No, sir, not yet.
Okay, so write down the tissues of the graft. Now you know what is a graft. The tissues of the graft become integrated with the tissues of the rooted plant, with the tissues of the rooted plant, and develop as a single hybrid plant over time. Develop as a single hybrid plant over time. Is it clear?
Yes, sir.
Yes or no? This is how what Fatima was asking. So last year during the same time, during the summers, there was a news which caught a lot of attention from India, from Uttar Pradesh. There was a man, like a farmer, who was doing this mango cultivation. He managed to grow more than 150 varieties of mangoes on the same tree. Anyone is aware of that news? And it actually, you can do that. You can grow more than one variety of mango on the same tree. Now, can you think how it would have happened? How it must have happened?
Different places attach different types of.
Yes. So this would not have been done like in one go. So if all of a sudden you make 150 grafts on a plant, it will be very overwhelming for the plant to take it, right? So over the years, he must have grafted certain different varieties on a trunk, on a mango stem. And over the time, as it must have got accepted, there is a new hybrid variety, new variety. So there are multiple varieties growing on the same root system. Make sense?
Yes, sir.
Yes or no? Yes. So this is how
Is that healthy?
healthy in the way, like you are talking from the perspective of nutrition.
No, like it is mixed, means it has different genes, like it has different people make watermelon, like pineapple and some other fruit, so mixing two fruits, is that healthy?
Yes. See, if you are not activating or if you're not imparting any toxicity to the plant, the healthy question, see, it's not natural. That is clear, right? But for that matter, everything that we eat right now is not natural. Everything, right from the cereals. So everything which came from wild, if you know how what wild cabbage looks like, all the cabbage and the derivatives like broccoli, cauliflower, and the cabbage, three most common things that came from the wild cabbage. You know, wild cabbage is very bitter and blackish in color, which is found in the wild, in the jungles, and you would not like to eat it as a vegetable. But then we domesticated, and with time we realized that there are certain varieties, certain mutants, after mutations, after certain varieties cropped up, that this is a better one. After cross breeding it with some other plant, we realized, oh, this is even better. And then we selected those which were better in taste and in nutrition. Similarly, for all our cereals, you know, there are more than 50,000 varieties of rice alone in the world, but we do not grow all of them. We only grow the best ones, which have nutrition, takes less resources. You know that paddy fields require lots of water.
Yes.
Rice farming requires lots and lots of water. The paddy fields are always full of. That's why they are cultivated during the monsoons when there is rain. But now with the water scarcity happening everywhere, is it possible to keep growing the same old varieties of rice which require lots of water? No.
Yeah.
Over time, genetically, like bioengineers means scientists, biologists have developed other mutants varieties, other genetically modified varieties which can tolerate less water. And by tolerate, I mean they can grow very well in less water. Their protein content is even more. So rice is a good source of starch and carbohydrate, right? But nowadays, the rice which we get from the market also has protein, especially in India. Farmers are advised and are directed to grow these specific varieties because India as a country started this revolution called Green Revolution. You know about this? Green Revolution. Yes, what was that? Making high yielding variety cereals and crops and full of nutrition, because everyone is not capable and not economically well off enough to get a balanced diet. So a balanced diet says that you need to also eat sources of proteins like egg and meat, sources of or dal, and sources of carbohydrates like wheat, rice, chapati, and then sources of vitamins and minerals, then milk, and all these things should be there. Then roughages, right? But most of the people cannot afford this diet, can they?
No, sir.
No.
Awesome.
So what is being these days, what is being done these days is that in a rice variety, rice is a good source of carbohydrate. But what if I have a variety of rice that also gives you some fraction of protein in your diet? Now this is very interesting, no? Or some
But eating protein and carbohydrates at the same time is not good.
no, our body needs everything at the same time because carbohydrates are the first fuel in the body. Glucose is the source of energy for each and every cell in our body, correct? Because glucose undergoes breakdown in the process of respiration with the use of oxygen, correct?
Yes, sir.
Same time, we also need amino acids and proteins to build, repair, and make our body, correct? Muscles, wear and tear, wound healing requires proteins. At the same time, we also need lipids and fats because all our cell membranes are made up of lipid. It is a lipid bilayer, correct?
Yes, sir.
At the same time, we also need vitamin and minerals because vitamins help in different physiological processes. Like vitamin A is good for your retina and helps in proper eyesight. Vitamin D is good for your bones and strengthen your bones. Vitamin C helps in some other things in the body, correct? You understand? So
Yes, sir.
anyways, we are eating everything at the same time. But if one food source can provide you more than one nutrient, is a good thing, right? Yes, it's not natural, but this is how it is. So it is healthy. Health wise, it is healthy. It is not a processed food. What is not healthy is a processed food, which by which I mean something which is deep fried, or something which is packed with preservatives, these things, or something which is heavily carbonated like cold drinks, soft drinks. They in a high quantity, if we consume, they are not good for health. Okay,
Yes, sir.
Fatima, so it's a good discussion, but you understood everything. Grafting. Now
Yes.
tell me, what do you think is the disadvantage of grafting? Quickly, write down disadvantages.
Sir.
This is a question that comes in your boards. Advantages and disadvantages of grafting. Yes, Arpit.
सर मैं तो क्वेश्चन आंसर कर रहा था.
Yes, yes, please answer करो.
Sir, it can cause infection.
To whom?
Sir, the grafting process, sir.
Yes, it cannot cause infection. It is more vulnerable to infections.
Yes, sir.
It is a vulnerable system. So first, yes, you can write it in disadvantages. First is vulnerable to infections. But suppose we cared for it, we took good care and there is no infection. But that is the only disadvantage. What do you
Short
think?
lifespan.
Yes, Kulsum?
Short lifespan.
Why? Why do you
Short lifespan.
Why?
Because
Yeah.
The seed propagated plants, in the bud plants, it is short compared to the seed propagation. The lifespan.
Like you mean to say that you mean to say that if I develop a plant through grafting, they will die faster?
Yeah, the budded grafting.
Okay, but isn't it an advantage instead of being a disadvantage?
How, sir?
Because see, if you germinate a plant from a seed, it will take first 2-3 years to become mature to be able to give fruits. Suppose you want to start a mango farming and you already have 2 mango plant trees, well developed 2 mango trees you already have. Okay, Kulsum? But you want to expand to other varieties. Now you have 2 options. One is what that man from Uttar Pradesh did. You start grafting different varieties on your 2 trees and start getting different other varieties as well. Or you take seeds of those varieties and plant it and start growing these new trees right from the beginning. So first 3 years at least will just go in the development of these trees so that they can start bearing fruits. No?
Yes.
So what do you think? Isn't grafting being a fast mode is a advantage? For us, it is an advantage, right?
Yes.
Yes. So any other obvious disadvantage? See, when we are doing vegetative propagation, you have to tell the advantage and disadvantages with respect to the method. Why are we doing it? What is the advantage or disadvantage in that? A normal mango plant lives for 70 years. The one you grafted died in 40 years. But that is not a disadvantage to you because you made profit. You made a lot of other varieties in that 40 years and you can very rapidly, before that plant dies, can make another hybrid through this process. You understand, Kulsum?
Yes, I understand.
If you were a mango plant, yes, it was disadvantages to you because you will be living 30 years less as a plant. Correct? So your
Yes.
answer is correct from a very different perspective. From a perspective of a plant, the plant would not like to become a hybrid, but because they cannot vote and they cannot tell what they want, we do whatever we want to do with them. So advantages and disadvantages with respect to the process. Anyone else can think of a disadvantage? See, I can tell you and dictate you, but the point is you need to learn to think through. More prone to infections, more vulnerable is one point. Second, don't you think it is a limited process? We cannot do it for all species of plants we want to propagate. Like cutting is even more widespread. You just cut a part, put it in soil. Here, you have to look for compatibility. You have to look for matching, and the plant without undergoing any infection should become a hybrid. So only few species could be grafted on some other few species, right? Let's say different varieties of mango can be grown on a mango tree. But can a papaya be grown on a mango tree? A papaya, banana be grown on a mango tree? If I graft a banana tree on a mango tree, can I do that?
No, sir.
No. So it is limited to matching species. You understand? So you can write down the second disadvantage. It is limited to us. It is limited to a fixed number of species. Grafting does not work well with all species that we want to propagate vegetatively. Any other disadvantage? Okay, any advantage? Tell me, what is the advantage? Why are we doing this?
We can get
Faster.
Sorry, Fatima?
It's faster.
Yes, it's a rapid mode. So you do not need to grow something right from the beginning. Like in case of cutting, we just put a stem in the soil and the stem did not have a root system. So we need to give plant growth hormones to develop the root system right from the beginning, correct? It will take some time. If you are germinating a plant from seed, that is another story. It will take years. But grafting is faster. Within months, a plant is ready to give fruits. Okay, yes, write down. It is a faster process to propagate, and you can make hybrids, correct? Hybrid plants and new varieties can be propagated. Second advantage. Is it clear to everyone? Tell me when you have noted down. Done? Fatima? Arpit?
Yes, sir.
Hafsa?
Yes,
Hafsa?
sir.
Done? Okay. Let's move forward. So from after grafting, we come to the third part, which is layering. Now, what do you understand by layering? Remember the runner stems I told you about? In the natural mode, just above, I told you about this, a runner stem. Remember here, through stem, so there is one plant, plant 1, that starts growing a horizontal stem, which is called a runner stem. It goes to some distance, and after that, it forms a node, a bud at the node, and a new plant, plant 2 arises. Now this new plant will also do the same thing. It will start sending another runner to some distance, then the third plant will come out, then a runner, fourth plant, fifth plant. You understand? So in a way, all these plants stay connected, but they propagate. There are very lots of individual plants made. Grasses are sometimes connected through runners. Lots of wild species of grass. Anyone has seen a grass in a lawn and you try to pull out one grass and 3 nearby grasses also come out? Anyone has seen it happening?
Yes, sir.
So if you try to pull grass in a park or a garden, sometimes some species of grass actually are connected through runners. So you try to pluck it out and you will see that a runner and then another plant also coming out.
Sir, how runner is also can be natural mode, right?
Yes, runner is a natural mode. We try to exploit this this technique through layering. Okay, layering is an artificial mode. So runners are formed by normally small plants, grass-like plants, or very bushy plants. But runners are not sent out by trees. Okay, but here we are trying to apply the same logic to trees. So there are many trees which are even like they're trees, but they're smaller, they are bushy. Fruit, especially fruiting trees. Okay, and what happens is, like if you see a pomegranate tree, anyone has seen a pomegranate plant? It does not go very high in the air, the stem. After some like a few foot, there are branches arising. So it's like a bushy plant. See, a tree is like this, a long stem, and then very up in the air, the branches starts, correct? But on the other hand, there are many fruiting plants which are like there will be a stem, short one, and then branches will start very early on. You understand what I'm trying to say? These are called bushy plants. Is it understandable or not? Tell me, then I'll move forward. Yes or no?
So
No? Okay, sometimes let me draw two trees. This is one tree, okay? Is it clear?
yes, sir.
This is another tree or a plant. Have you seen these two type of plants around you? In one example, the branching which is happening on the stem is happening at a very high distance. So let's say after 3 meters or something. But on the other hand, just after, let's say, 1 foot, there is branching that started. These type of plants are called bushes. Have you seen bushes?
Yes, sir. Yeah.
People use to make boundary or fencing around their properties also.
Yes, sir.
What kind of plants do we use to make fencing or boundaries, which are very bushy, like which starts throwing branches after few inches above the ground, correct?
Yeah.
Then only it makes sense to make a fencing with that, because the fencing is to avoid animals coming in or anyone coming in, correct? But if you just grow trees in a line, will it make a fence?
No, sir.
Because there is space here. Branching is happening very up. Branching is happening here. But anyone can move in between. Is it clear now?
Yes, sir.
Plants like this, branching starts happening very low towards the ground. Okay? Many fruiting plants are like this, in which branches starts arising very, or like very low towards the ground. Now is it clear?
Yes, sir.
All? Hafsa? Say yes or no?
Yes, sir.
Yes. Okay. So now let me move forward. So in layering, what happens is we take such a plant in which there is a branch which comes up early towards the ground. This is one branch. This is another branch. Okay. And we try to pull this branch down because it has to be flexible also. So you try to pull this branch down, and we put this branch in the ground and leave a part out. You understand? And then we cover it with the soil. You understand? So it's like in a plant where branches are arising low towards the ground, you can just pull one branch down, dig a hole in the ground, and put one part of it inside and put soil on the top. So that is called. Do you understand what I have done here?
Yes, sir.
Branch is still attached to the plant, but now some part of it is inside the soil. Now what will happen, the part which is inside, after some time it will start developing adventitious roots. Okay, and this shoot part will start developing branches of its own. You understand? And from this one plant there will now be a second plant nearby, because it has its own root system. This will also develop. Now this root should not have been developed at this place, right? So it is called adventitious. Is it clear? Because now it's inside the soil and it will induce root formation when it is inside the soil. Is it clear or not? And this is
Yes,
the
sir.
new shoot that develops. So this was supposed to be a branch. After some time it became a whole new plant, and this new plant is called a layer. The newly developed shoot with adventitious roots is called a layer. So for plant 1 there is one layer. Now when these two plants are growing side by side and they have even more flexible branches, you can pull all these branches and put them in the soil, and you can have multiple plants around. You can make a group of plants. Make sense? Is it clear?
Yes, sir.
Isn't the concept same like a runner? Look here. But that was happening naturally. The plant itself was sending something out which was developing roots. Yes or no?
Yes, sir.
Yes, sir.
They're artificially exploiting that concept for higher plants, for bigger bushy plants. Is it clear to all of you? Say yes or no.
Yes, sir.
Wholesome? Afsa?
Yes, sir, I understand.
Okay, now write down about layering. In layering, the stem or branch, in layering, the stem or branch of a plant is bent to the ground, is bent to the ground, and a part of it is covered in the soil. And a part of it is covered in the soil. Next point, the part covered in the soil develops adventitious roots over time. The part covered in the soil develops adventitious roots in time or in some time. And the third point is the attached branch or stem along with the newly developing adventitious root, along with the newly developed adventitious roots is called a layer. Is called a layer. Okay. Therefore, this process is called layering. Is layering clear, everyone? Any doubts? In layering, grafting, or cutting? Any doubt is there? Let me know when you are done writing, or if you want me to.
Yes, sir. I'm done writing and no doubts.
Excuse me, sir.
Anyone wants? Yes, please.
Sir, when the second plants grows its roots, so sir, the connecting parts that is connecting the both the plants, sir, does it break over time?
It may or it may not, because ultimately after some time it is a, even if it is a connected system, what basically we do, what the cultivators or the farmers do, they sever it themselves. So they take an axe and cut the part, but it may not sever on its own. You understand? It will not break on its own because it's a live part, and now there are two active root systems which are taking nutrition from the ground. So you know, it's like a double-decker plant with two sources of nutrition. But sometimes the cultivators do not want it, because if one plant gets a disease, the other may also get it. If one plant is not making good fruits, the other may also not make it. So when the second layer is developed enough that it can now sustain as a new plant itself, and how can you make sure of that by looking at new branches which are forming. So when you make a layer, in the beginning it is just the tip of that branch which is outside the soil, right?
Yes.
If that layer has started developing roots, then you will see certain changes in that new branches and new things are forming, and it is taking a shape of a new plant on its own. So you sever the link which was in between, because now it's not required. Is it clear?
Yes, sir.
Yes. But plant will not like just kill it by itself, because plant is not aware of what we are doing to it. No? Plant does not know. Okay.
Yes, sir.
Okay. So if it is clear, let's go to the last part for today that we'll be covering, and it's tissue culture. Tissue culture. Now, to understand tissue culture, as the term suggests, we take some tissue, and what does culture means? We are culturing it in artificial environment, right? We are making it grow. But why are we studying tissue culture in reproduction? Because in plants, it is possible to take a tissue and grow a whole plant out of it. Okay, or at least a plantling which can be put in the soil later on. So in tissue culture, what we do, first let's write down some things about it. In tissue culture, plant tissue from some specific parts of the plant tissues from some specific parts of the plant are cultured in the laboratory setup, are cultured artificially in the laboratory setup in order to develop a new plant. In order to develop a new plant. Okay, now I will take you back to junior classes and I'll ask you, in your junior classes you have studied about meristematic tissues, right? Meristematic tissues, people?
Yes, sir.
What is it?
Sir, it is present at the tip of the plant.
Apical bud and the apical bud, lateral bud, right? The apical meristematic tissue, the lateral meristematic tissue. So if this is a plant, this is a stem, these are branches, that then at the tip of it and also at the branches. So some tissues present here and some tissues are present here. So these are called apical meristematic tissue, and these are called lateral meristematic tissue, correct? Apical helps in the elongation of the length of the plant. Lateral helps in the increase of the width or the girth of the plant. Remember? Class 10th, 11th. Say yes or no? No? No one remembers meristematic tissue? Okay, so write down a in-class assignment which you have to do. You have to go back and find out different types of plant tissues that you have studied till now. Okay, these are going to come in entrances. I can just assure you of that. So what are meristematic tissues and what are other types of tissues? You have to read. Everyone has written the assignment?
Yes, sir.
Everyone. Kulsum, Fatima, Hafsa, Meraj.
Yes, sir.
Yes, sir.
Okay. So meristematic, let me tell you, meristematic tissues are those tissues, and if you remember while I am telling you, please let me know that yes, you remembered. Meristematic tissues are actively dividing tissues of a plant. And only these tissues have the ability to keep dividing and form new structures, form new buds, form new branches, and form new leaves. These are called meristematic tissues. Often for tissue culture, we take tissues which have the capability of actively dividing. Why? Because if it will not divide, then we will not be able to propagate it into a whole new plant. Yes or no?
Yes, sir.
So write down the tissue used in the tissue culture process. The tissue used in the tissue culture process must have active dividing capability. Active dividing capability. So what happens is we take a tissue, okay, from a plant. In laboratory, we provide this tissue with plant growth hormones. Of course, without plant growth hormones, we will not be able to induce different structures like roots and stem, etc. Correct? We also provide this tissue with nutrition, because right now it will not be able to make food for itself because it's not a plant yet. And tissue is made up of multiple cells, so I'm making different cells here. Is it making sense to all of you?
Yes, sir.
After some time, this tissue develops into something called callus. Callus is a structure, like it starts taking some shape, which later on starts developing into active root-like and shoot-like structures. Okay, this is called a plantling or small plant. Then you take this and you put it in soil. Again, in laboratories only, under controlled conditions, where in the soil they start developing proper roots and proper shoot. Is it clear? Now, this is a proper plant, and once it reaches this stage, then we plant it wherever we want. Plant in soil outside the laboratory. Is it clear, the process?
Yes, sir.
We started with just tissues, which were just a bunch of cells, but we developed them by plant growth hormones and nutrition into callus, then into a plantling, then into a proper plant, and then we can propagate it into the natural environment.
So how much time it takes?
It depends which plant you are culturing through tissue culture. So it takes time, but why are we doing it is the question. Can you think of why it is? Yes, Arpit.
Sir, we can develop any plant by this tissue culture.
But why to do such expensive thing for any plant? Why would you like to waste so much of money in laboratory equipments, bringing plant growth hormones, nutrition, taking care of
Research.
just for research? If it is done, then it's not vegetative propagation, it's plant research.
Oxygen.
We have lots of oxygen. Just one small tissue developing into a plant cannot give us oxygen to survive. No? We have forests. If we want oxygen, we will not do deforestation, which we are actively doing. Any other thing? See, when all the doors must have closed, when cutting is not working, grafting is not working, nothing is working, seeds are not an option, then we might go for tissue culture, correct? Yes or no?
Yes, sir.
Clearly, this technique is helpful in propagating the rare plants, the plants which are endangered in the wild, which are about to go extinct, like a purple orchid. So there are many species of orchid. You know orchid? Orchid is a flower.
Yes, sir.
Very rare one. You will not find orchids, all sorts of orchids everywhere. So there are many rare plants which have value but are going endangered or extinct. So through this method they can be propagated. So write down, tissue culture technique is helpful. Tissue culture technique is helpful in increasing the number and propagating the endangered plant species or the plants that are unable to form seeds and grow under natural conditions, that are unable to produce viable seeds. Include the word viable, which means not useful, which are unable to produce viable seeds and grow under natural conditions. So this is why tissue culture is done. But again, it is a mode of vegetative propagation. It is a mode of asexual reproduction, because here we are from a tissue making a whole new plant. We are making copies of a plant, correct? But it will be genetically identical to the plant from which we took the tissue. Yes or no?
Yes, sir.
That's why it's asexual. So this is where we end asexual mode, and from next class onwards I'll be starting with the sexual mode. So let's first finish with the plant sexual part, which means sexual reproduction in flowering plants. Okay, we'll take that first, then we'll go to humans and animals. Is it clear? Is it okay?
Yes, sir.
Kulsum, could you understand everything that was taught today in the class?
Yes, sir. Understood.
Okay, so for the topics which you missed, please look for the videos, find get your hands on those, and we will figure out for your extra classes. Okay, then see you in the next class, everyone. Please keep revising, and for the next class, before you come, because there is a week in between, so I am giving you a homework. You have to read 3 things. Read about flowers, their structural anatomy, what are flowers, what other different parts they have, and read about seed. What is a seed? Okay, flowers and seeds. This will help in our discussion in sexual reproduction of flowers, of flowering plants. Okay, everyone.